Well, let's start today the next part of last topic. In the last video, we have seen a monohybrid cross, and obviously now you should understand what is monohybrid. Now we'll move ahead with a dihybrid cross, and in this dihybrid cross, we will learn and understand the two laws of Mendel, that is law of segregation of gametes and the law of independent assortment. Okay, so these are the two laws which we are going to justify by the example of dihybrid cross. Now in dihybrid cross, the term di, why it is here, that is why this di is here. Di means two, just like mono means one, di means two, and hybrid, that means here the combination is formed by two different traits which are alleles of each other. Now, obviously, you will now have to understand what is allele. See, the traits or the characters are this one. So this is homozygous, round and yellow. Remember, this is written in capital, so it is dominant trait. Now these are the allelic forms of it, means the same gene but codes a different trait, slightly variable trait in case of the shape and in case of the color. So round is the shape and yellow is the color of these two genes, and here this wrinkled and wrinkled and green, these are the two allelic forms. Now what is going to happen in F1 generation or first filial generation? We are getting this R and this R, this Y and this Y in the combination, and what it will produce? It will produce a hybrid with two traits, round with this wrinkle, yellow with this green. That means this is hybrid in two traits or character. Now why it is hybrid? To understand this, just take one thing in the mind, that where is the location. This R is on one chromosome of one parent, and this Y is on one chromosome of one parent. In the same way, this small r, which is coding for wrinkled nature, is present on chromosome of another parent. Likewise, this Y, which is coding for green instead of yellow color. So these are present on different chromosome. They are going to unite after the gamete union. So here the laws are being formulated based upon the meiosis. So in meiosis, what happens? Gametes are formed. In this homozygous, round and yellow, these are the gametes. R round, Y yellow. So these are the gametes. Why they are double? Because this is a diploid organism. Plants are diploid, so they have a double copy of each gene, and these diploids becomes haploid when they forms the gametes. So these are single of their gametes. Likewise, for this wrinkle and green, you will get one small r, small y, small r, small y. Okay. Now, what is the first law is being depicted here? The law of segregation of gametes. That means these gamete separate, and each comes inside the gamete 50%. They were 100% here, but in gametes they will come 50% from one parent's gamete. Other 50% comes from this parent, and these two 50% combine together to form the 100%. Now here, since we understood that these gamete genes are present on separate chromosome, so what is happening? The genes from different chromosomes are uniting together to form this hybrid, and these two different traits which are present on two different chromosomes are going to be expressed in F2 generation. All are not expressed in F1 generation. So in F1 generation, remember, only the dominant express, the recessive never express. This is universal in F1 generation. So what we should conclude, that see, we got a dihybrid, the homozygous round and yellow, homozygous wrinkled and green. After meiosis, gametes are formed. Then the two parents' gametes unite together, that we express as a simplification by Punnett Square. Okay. Now on this checkered board you see parents 1, that is female parent, we consider, and the male parent. Now here we have arranged the gametes. Simple way, the gametes are written on the outside of the table. Now see how these combinations are there. 1R, this is from female parent and this R is from male parent. They unite together for one character. This Y from one, that is male parent, and this Y from female parent, it come and unite together. In this way they make a dihybrid, that is two traits with their allelic form. But in this F1 generation only the dominant trait expressed. Recessive traits do exist but they are not expressed, they are being suppressed by the dominant character. So like the way you have prepared the checkered board or Punnett Square for monohybrid cross, you just have to add these gametes together. See, here you added, it becomes like this. Here you add, this R comes here, this R comes here, this Y comes here, this Y comes here. In the same way, this R comes here, this R comes here, this Y comes here, this Y comes here. In the same way, this R comes here, this R comes here, this Y comes here, and this Y comes here. What is unique here, that in F1 all only dominants are expressing, the recessives are not expressing. So if this continuously go on, then you will not get the new characters or variation. And to have that, the nature has prepared the second law that Mendel considered as law of independent assortment. What does this law of independent assortment is going to explain? That these two traits with their hybrids, 50% they came together by segregation and unite during fertilization between two different parents. Now these two traits separate independent of each other, and by that we'll get some combinations which were not present in F1 generation. What we are getting in F1 generation, all are round and yellow. Remember, the parent was round and yellow but it was homozygous. All F1s are round and yellow but they are heterozygous. That means the dominant R with its allelic form small r, that is wrinkled, is present here. So this is law of segregation by which gametes are formed. These gametes 50% comes in each what you call parental genetic combination. These unite together to form 100%. Now here these are hybrids for two traits. Now what we are doing to justify law of independent assortment, that how independently new combinations occur or how gametes segregate by influence, not getting influenced by effect of one gene with its combinant gene. You see here in F1 generation R is expressed with Y. So these are the showing togetherness. But in case of F2 generation we are going to get some new combination. How? All F1s are round and yellow, but in F2 you are going round with green or here wrinkled with yellow. These are not the combinations were present in F1. How they are getting? Just see here. What we are doing now? We are now crossing or selfing the F1 progeny. Okay, like for monohybrid cross we have done. So we have got the gametes, of course. The gametes are formed. Now here, since it is heterozygous, you will get instead of 2 2, you will get 4 4 gametes here. R Y R Y. Okay, small r, capital Y, small r, small y. In the same way, you will get here also. Simple. The gametes here 2 are formed. Y here 2 are formed because these are homozygous. Y here 4 are formed because this is heterozygous. Okay, this is heterozygous for two traits. That's why we are getting 4 different gametes. So law of segregation, that is gametes 50% segregated. Now what we are going to justify? Law of independent assortment. That means the genes will express independently of their another gene. Here both these genes are expressed, but by independent assortment we are going to explain that this round always does not comes with yellow, neither this yellow does not comes with round. Some new combinations or variations occur. To justify that, just go to the Punnett Square with 16 dimension. What we have to do, keep one parent's gametes at one side and another parents at another side. Say this is male gamete, this is female gamete. What we are going, just add the gametes together. Okay, this R, this R, this Y comes here. In the same way we'll move, this R come, this R come, this Y comes, this Y comes. In the same way when you go and add these gametes, for example randomly I come here, small r, capital R, capital I. Now the question comes, why this R is not first here? As I said in the previous video, that the dominant is being kept at the first place. Okay, you can come at this position and locate from where it is got. See, this R is from here and this R is from here, this Y is here and this Y is here. Okay, randomly you can go anywhere. For example, you come here, small r, capital Y, small y. See from where this came, small r came from here and small r came from here. Now the capital Y came from here and small y came from here. So randomly wherever you go, it is just addition of the gametes of two different parents. 50% of one parent, 50% of another parent. Now, as I said, in law of segregation we got gamete formation and 50% chances to participate the trait into one gamete. Okay, here in law of independent assortment, remember what we are going to justify. We are going to justify that this round does not always express with Y, or this yellow does not always express with the round. They segregate independently and they express. How? See, I have written here RG, that is round green. Round green was not any combination in the homozygous parent, either dominant or recessive, or even in F1 generation. See, round green was never there. Round is the first dominant trait, yellow is the second dominant trait, wrinkled is first recessive trait, and green is second recessive trait. So this round and green is nowhere here in the parental or in F1, but in F2 we are getting round and green. In the same way we are getting wrinkled and yellow. Nowhere here you will find wrinkled and yellow. You will get round yellow or you will get wrinkled green. But here this wrinkle with yellow is a new combination, and that is what the application of second law of Mendel, that is law of independent assortment, which justifies that these traits express, or these genes express independent of other, and that's why it is called as independent assortment. And what is application of this? In variation formation or new character formation is the application of this independent assortment. So what new we got? What we expected? Round and yellow, they are dominant, all expressed in F1 generation. Wrinkled green, since they were recessive, they will not express in F1 generation, but they do express in F2 generation. What is independent assortment of gamete? That these two dominants are not always together. They express independent of one another with their allelic form. That's why we got round green. That's why we got wrinkled yellow. So these are the two new combinations which were not present, and this is because of this second law, law of independent assortment. New combinations, new variations are formed. So at the end of this checkerboard, I hope you understood how to add the gametes. One parent gametes, one side, say male, female, on the upper side, just add. Randomly, once again I will say how this capital R, capital Y, small y came. It indicates round and yellow in heterozygous condition. See, this R came from here, this R came from here, this Y came from here, and this Y came from here. In this way, you can just add the gametes, one from each parent. Okay, so at the end of this gamete, what you call union, you will get this ratio, 9:3:3:1. Now what does this 9 indicates? This 9 indicates this 1, 2, 3, 4, 5, 6, 7, 8 and 9. These 9 indicates both the dominant trait exists there. So these 9 are round and yellow, both dominant trait expressed. Okay, so this 9 indicates expression of both the dominant gene. Okay, this 3 indicates round and green. That means first dominant and the second recessive is here. So these 3 indicates first dominant and second recessive. Now in case of these three, wrinkled yellow, this here second dominant exists and first recessive exists. See how the logic is. Round and yellow, both the dominant trait expressed in 9. Round and green, the first dominant expressed in these three. Wrinkled yellow, the second dominant expressed in these three. And wrinkled green, as usual, the homozygous, that is being suppressed in F1 because of homozygous condition. It expressed. How this RRYY comes? Simply see. This R of one parent, this R of one parent, this Y of one parent, this Y of one parent. In this way, it has got both the homozygous, what you call union of characters or traits, and it expressed because there is not a single dominant gene here. That's why it expressed in F2 generation, and that is the speciality of this recessive character. They are going to express in F2 generation. They are never going to express in F1 generation, since in F1 generation they are in heterozygous condition and always a dominant allele present there, so that suppress their expression. Now this is the phenotype, that is the exterior or physical appearance. Actually what genetic combination? See, you can see here homozygous round and yellow, you can see here homozygous wrinkled and green, but here it is not the exact 100%. The roundness is homozygous, but the yellowness is heterozygous. So in this way, the genotype is not exactly the same as phenotype. The genotypic ratio is different from phenotypic ratio. Okay, so we know the phenotypic ratio 9:3:3:1. 9 indicates both the dominant trait. First 3 indicates first dominant trait, that is roundness. Next 3 indicates second dominant trait, that is yellowness, and the 1 indicates the homozygous recessive parental combination, that is wrinkled green or small r, small y, small y. Genotype is different. Now to mug up genotype, what we have to do, just this is casual, we can remember the dihybrid F2 generation ratio 9:3:3:1, but its genotype is simple. What we have to remember? We have to remember just one number, 1 to 1, just like Airtel helpline number. Okay, 1 to 1. What you have to do? Double this number. 1 to 1, 242. You got 1 to 1, 242. What was the number you doubled? 1 to 1. So what is the number of F2 or the genotype of dihybrid cross? That is 1 to 1, double it 242, then single it 1 to 1. So simple. 1 to 1 उसी को double करो 242, उसी को single करो 1 to 1. That means the genotypic ratio is 1 to 1, 242, 1 to 1. That's it. 1 to 1, 242, 1 to 1. 1 to 1, 242, 1 to 1. In this way, you can mug up the genotype in F2 generation of this dihybrid cross. So what we justified today? Law of segregation of gametes, that is gametes segregate and each parent spend 50% of their what you call genetic combination. And second, law of independent assortment, because of which these two genes express independently of each other. That's why we got the new combination round green and wrinkled yellow, which were nowhere present in the parental or F1 generation. Round yellow, wrinkled green. There were nowhere round green and wrinkled yellow. And because of this independent assortment of gamete, new combination comes to existence, which is the raw material as the variation for evolution. So what is the end point? To remember law of segregation, the gametes segregate, every parent donate 50%. Second law, law of independent assortment, where one gene express independent of another, and because of that new combinations occur, and these new combinations are the variations that brings the evolution to come into existence. Now next, the dihybrid phenotypic ratio 9:3:3:1, and dihybrid genotypic ratio, simple number 1 to 1, double it 242, single it 1 to 1. That is 1 to 1, 242, 1 to 1. In this way you can understand the chemistry of this dihybrid cross. So I wish you understood it correctly. Read in the NCERT once twice, you will definitely come to the conclusion. Law of segregation and law of independent assortment with its genotype and phenotype ratio. So I hope you understood it. Thank you. Thanks for watching.